Okay, so any doubt? We're moving forward now and we start the real metabolism part. Okay, this is an overview of every reaction that you're going to study in the later pages. Okay, this is a glycolysis thing. You don't need to remember every step in the glycolysis, but some rate limiting steps and some other important steps. Okay, you should remember about them, like the conversion of fructose 6-phosphate into fructose 1,6-5-phosphate. This is a rate limiting step you will study, and similarly other steps also. This last step you have to remember. Okay, this is the glycogenolysis and the glycogenesis part. How it will happen, you will study that later. Again, the HMP shunt, the fructose metabolism and the associated diseases, the galactose metabolism and the associated diseases we will study. Similarly, the gluconeogenesis, which is going this way. This is a glycolysis. Okay, then the formation of lipids and the lipid metabolism, the formation of ketone bodies, the protein metabolism. Remember the methylmalonic acid, the vitamin B1 thing. Okay, the TCA cycle and the urea cycle. All of these things you're going to study in the later part. Okay, so yeah, we will move forward. And you should remember how much ATPs are produced after a single molecule of glucose is breakdown. Okay, should we remember this fact that 32 net ATP is formed in the heart and the liver because they require this malate aspartate shuttle. But in the muscles, it is 30 ATPs, not the 32, but the 30 ATPs actually, because the muscle uses the glycerol 3-phosphate shuttle. It is a fact, you should remember it. Okay, and in the anaerobic glycolysis, because in the anaerobic glycolysis, you are just converting all of your pyruvate into the lactic acidosis, but not the acetyl coenzyme. So you will get only the 2 net ATPs, not the 32. Okay. You should also remember the fact that the metabolism of 1 gram of protein actually produces 4 calories of energy, and 1 gram fat will produce a 9 calories of 9 calories of energy. You should remember these two facts. They will ask you to calculate it if a man or a female requires this amount of energy, how much of the energy should come from the protein or come from the fat or come from the other substrates. So you should remember that 1 gram of protein actually forms a 4 kilocalorie and 9 gram of fat forms a 9 kilocalorie. Okay. You should remember about the function of these carrier molecules and what they carry in the activated form. The ATP carries a phosphorylated group. Whenever the ATP energy requires, it will convert into the AMP and ADPs, right? So it carries the phosphoryl groups. All these NAD, NADPH, FAD, FADPH, the FAD, H2, FMN, all they carry is the electrons, which will ultimately requires an electron transport system that you will study in the mitochondrial sections. The coenzyme A and the lipoamide, it will require acyl groups. They carry the acyl groups. The biotin, which is a vitamin B7, it carries a CO2. And that's why I told you guys that all the carboxylation reactions requires this biotin. The tetrahydrofolates, all they carries a 1 carbon units, which will be utilized in the formation of this, thymine. Okay. The SAM molecule I told you, or the methyl reaction, the imprinting, the DNA methylation, the histone methylation, all of these reactions require this SAM molecule because it carries a methyl group. The TPP, this thiamine pyrophosphate, the vitamin B1, it carries the aldehyde group. Okay. And you should know about the function of this NADPH. It is involved in the anabolic processes, like the formation of your fatty acids, the formation of cholesterol, the formation of steroids. All of these reactions require the NADPH. Similarly, the respiratory burst, which takes place in the neutrophils for the generation of reactive oxygen species, it requires the NADPH. The cytochrome P450 system requires the NADPH. The glutathione reductase enzyme, which is involved in the, in neutralizing these reactive oxygen species, it requires a glutathione reductase. Okay. Sorry, it requires a NADPH. Okay. So as I told you already in the previous lecture, that NAD is generally used in the catabolic processes. Remember, when I was talking about the vitamin B3, I told you that NAD is involved in the catabolic processes and NADPH is involved in the making processes or the anabolic processes. Should remember these processes only, the anabolic processes like fatty acids, steroid, cholesterol, the respiratory burst, the cytochrome P450, and the glutathione reductase function. And now you should know about the very first enzyme of glycolysis, which are hexokinase or the glucokinase. You should remember their location of these two enzymes because the glucokinase is found only in the liver cells and the beta cells of pancreas, whereas the hexokinase is found in every cell of your body except these two cells, the liver and the beta cells of pancreas. Okay, then you should also know about this Km value of these enzymes, which shows the affinity of these enzymes. You should know that the hexokinase has so much affinity for the glucose. So whenever a glucose is available, these enzymes will get activated very instantly. So they will have very high affinity, and that's why a low Km value, you will study about this Km value later in the pharmacology section. Okay, so lower means very high affinity, and the glucokinase has very low affinity for it. And that's why whenever you have very high load of glucose inside your body, only then these enzymes will get activated. This glucokinase enzyme will get activated at very high glucose level. Okay, then the Vmax, these enzymes have very low speed, low capacity, but glucokinase has very high capacity. Okay, it acts very rapidly once they get the glucose. So hexokinase cannot be influenced by the insulin, but this glucokinase is influenced by the insulin actually. And that is a fact, you require the insulin to store the glucose in the liver, right, to form the fatty acid inside the livers. So this glucokinase is affected by the insulin, but hexokinase do not affected by the insulin. You should know the substrate of these, of what substrates give them the negative feedback. Remember this step, the glycolysis, the conversion of glucose into the glucose 6-phosphate. It could be done by one of the two enzymes there, the glucokinase or the hexokinase. But the glucose 6-phosphate actually gives a negative feedback to the hexokinase. And the fructose 6-phosphate gives a negative feedback to the hexokinase. Sorry, to the glucokinase. Okay. Glucose 6-phosphate gives the negative feedback to the hexokinase, and fructose 6-phosphate gives a negative feedback to the glucokinase. You should remember this one. You should also need to remember this hexokinase can convert either the glucose or the fructose or the galactose even. No, not the galactose, I think, or the fructose and the glucose. They both can be converted by this hexokinase because it converts a hexose sugar. But the glucokinase can only convert the glucose. You require a separate fructokinase enzyme in the liver to convert the fructose into fructose 6-phosphate. Okay, or the fructose 1-phosphate, not the 6-phosphate, but fructose 1-phosphate. And you should remember this hexokinase actually converts the fructose into fructose 6-phosphate, but the fructokinase converts the fructose into fructose 1-phosphate. That is the difference between these two enzymes. Okay, so remember the function of this hexokinase, not the glucose only, but the fructose also. Okay, any doubt? Any doubt? And yeah, please remember this this disease known as a MODY, which is a maturity onset of diabetes of young. And in this disease, you will get a deficiency of glucokinase. And so that only your liver cells would be affected. So you will get a very, very mild diabetes in these patients, which do not require any kind of drug or anything. They have just mild baseline diabetes in them because of no function of this glucokinase. Okay. So any doubt, guys? I'm moving forward now. Okay, then the key regulatory enzymes or the steps in the glycolysis, you should remember. The first thing is the net production of ATP or the net production of substrates that's going to happen in the glycolysis. So glycolysis requires a glucose, two phosphates, two ADPs, two NAD. Remember this, and this reaction is not balanced. Okay. Please note that this reaction is not balanced. Okay. So you require two phosphates, two ATPs, two NAD, and it will form ultimately two pyruvates. From one molecule of glucose, you will get two pyruvates. You will get two ATP production. You will get two NADH production, and the formation of two water molecules and two hydrogens. And like I said, if you get, as you can see here in this reaction, yeah. Remember I told you after the glycolysis, when the pyruvate is formed, it could convert it into the lactase, and it requires, in the lactate, and it requires this NADH, all right? And then I told you, in case of this anaerobic glycolysis, what you will get is only the net two ATPs, because in the anaerobic glycolysis, all your pyruvate is being converted into the lactate, so that the two NADH that forms will be completely utilized to form the lactase, the lactate, and you will get only the net 2 ATP generated after the anaerobic glycolysis. So you understand it now? And then the key regulatory steps in the glycolysis. The first is the conversion of glucose into glucose 6-phosphate. Like I told you, one of the two enzymes can be used in this process. If it is liver or the pancreatic beta cells, it is glucokinase. In every other cell of your body, including the RBCs, it is your hexokinase. Okay. Glucose 6-phosphate gives the negative feedback to the hexokinase, and fructose 6-phosphate gives the negative feedback to the glucokinase. Then the next step, which is the conversion of fructose 6-phosphate to the fructose 1,6 biphosphate or the bisphosphate, it requires an enzyme known as a phosphofructokinase 1. Like I told you, it is a rate limiting step. There is one more enzyme, which is a phosphofructokinase 2. Let's come down here now. As you can see, this is a fructose 6-phosphate, and it could be converted into the fructose 1,6 bisphosphate with the action of this enzyme phosphofructokinase 1. And during the gluconeogenesis, this fructose 1,6 biphosphate would be converted into the fructose 6-phosphate by the action of enzyme fructose biphosphatase 1. Both of them are the rate limiting enzymes in the separate in their respective reactions. Okay. And now there is another enzyme which is a phosphofructokinase 2, which uses this fructose 6-phosphate to convert it into the fructose 2,6 biphosphate. And as I told you that this fructose 2,6 bisphosphate is an activator of this enzyme. It induces the enzyme to work more, as you can see. This is a fructose 2,6 biphosphate, which is the activator of this, inducer of this step. Okay, now what happens is, and yeah, similarly, you have this the fructose biphosphatase 2, which converts the fructose 2,6 biphosphate into the fructose 6-phosphate for the gluconeogenesis. So in this way, you are having this glycolysis, and in this way, you are having this gluconeogenesis. So what enzymes or what proteins are affecting this part? So whenever you are in a fasting state, what happens is your glucagon will releases, and with the help of this cyclic AMP, what it will do, it will activate the protein kinase A, and this protein kinase A is further activates this this fructose biphosphatase 2. So now you're removing this fructose 2,6 biphosphate, right? And now this fructose 2,6 biphosphate will no longer be able to induce this enzyme. So you are decreasing this step. That is the first thing. And secondly, in the fasting state, you are activating this fructose biphosphatase 1. So you are getting more gluconeogenesis and less glycolysis, because you are inhibiting this enzyme or activating this enzyme by the action of glucagon. Okay. Similarly, in the insulin, in the fat state, your insulin will decrease the cyclic AMP. It will decrease the protein kinase A activity, and as a result, this fructose biphosphatase 2 will get inhibited, and the activation of phosphofructokinase 2 will happen. So you will get more fructose 2,6 biphosphate, which will induce the function of this phosphofructokinase 1, and you will get more glycolysis and less gluconeogenesis. So this is very important, more glycolysis and less gluconeogenesis. In fed state, and in fasting state, less glycolysis and more gluconeogenesis. So that's how your glucagon and insulin control this step. They do not control the phosphofructokinase 1 or the fructose biphosphatase 1 directly, but the indirect way actually by the action on this substrate, the fructose 2,6 biphosphate. That is the actual thing. Okay. Any doubt in it? It is very important, the role of glucagon and the insulin on this pathway. Just remember the fact that you don't control these enzymes directly, but through the action on this fructose 2,6 biphosphate. Okay. And that's why, the first aid do not mention anything about the glucagon or the insulin on this step. Just control the fructose 2,6 biphosphatase. So guys, any doubt or should I move forward now? Please respond.
Move forward.
I can repeat this. Yeah.
Move forward.
Okay, then another very important step, which is a conversion of 1,3-biphosphoglyrate into the 3-phosphoglycerate, which is this step. Let me take you back. Yeah. This step, conversion of 1,3-biphosphoglyrate into the 3-phosphoglycerate. And you should remember this step because of the RBCs. Okay, so what happens is there is a mutase enzyme, which sometimes does what is, in the conversion of this 1,3-biphosphoglyrate, this mutase enzyme converted into the 2,3-biphosphoglyrate and then to the 3-phosphoglycerate. Okay. And what happens is there is a loss of ATP happens because this step generates 2 ATPs because it happens 2 times. So it generates actually 2 ATPs, right? 1 time 1 ATP. But if you convert this if you convert this 1,3-biphosphoglycerate into the 2,3-biphosphoglycerate and then to the 3-phosphoglycerate, then you won't get any ATP formation. Okay, and this is a very crucial steps that take place in the RBCs. And you should know that this 2,3-biphosphoglycerate actually help in losing the oxygen from the hemoglobin, releasing the oxygen from the hemoglobin at the tissue level. So this mutase enzyme is especially activated into the tissues to release the oxygen from the hemoglobin. And in this way, you won't get any ATP generation. So what happens is sometimes when your RBC is in the lung, they will get net 2 ATP production. Remember, in the RBCs, there is no mitochondria, there is no nucleus. So there is no TCA cycle takes place. All they will get is the formation of lactic acid. No? Remember? They will get only this pathway. They will get only this pathway, this pathway, not this pathway. So they will get a net 2 ATP production always. But because in the tissues, there is the conversion of this 1,3-biphosphoglycerate into 2,3-biphosphoglycerate, there is no net ATP production happen. So some sort of STEM might ask you something about that, like, there is some kind of a cell which is sometimes getting a 2 ATP, a net 2 ATP, but sometimes they're getting 0 ATP. So how this is possible? Because these are RBCs and they have this mutase enzyme. Please remember the enzyme name and its function. Sometimes you will get 2 ATP, sometimes you will get 0 ATP only. And this is a special thing with the RBCs. Okay, then the final step, which is a conversion of phosphoenol pyruvate into the pyruvic acid. Okay, and this enzyme requires a pyruvate kinase. You will again get 1 ATP from this step. And because this step takes place 2 times, so you will get net 2 ATP. So 2 ATP from here and 2 ATP from here. Okay, a total of 4 ATP will form, but 2 ATP is utilized in the initial step, which is these steps. You will require 1 ATP in this step and 1 ATP in this step. Okay, and you will form 1 ATP in this step and another ATP in this step. So a net of 2 ATP production happens. Understand what I'm trying to say? I told you, you utilize this 1 ATP here and 1 ATP here. but forms 1 ATP here and 1 ATP here. And then I'm saying you will get a net 2 ATP production. How is this possible?
That lower cycle take place 2 times.
Yeah, exactly, because these cycles take place 2 times. Okay, please remember this fact. Okay, then comes your pyruvate dehydrogenase complex. Okay, so dehydrogenase, like I told you, it requires the this thiamine. All the dehydrogenase reactions require this vitamin B1. It requires the lipoic acid. It requires the coenzyme A. Like I told you, B5 is the coenzyme A actually. B2, the riboflavin is the FAD. B3 is the NAD. Okay, so it requires the vitamin B1, vitamin B2, vitamin B3, vitamin B5 for the pyruvate dehydrogenase complex. And this is a complex that converts ultimately your pyruvate into the acetyl coenzyme A. Okay. And sometimes step one gonna ask you what is another complex which requires all the cofactors exactly like the pyruvate dehydrogenase complex. What other complex require all these 5 cofactors? So you should remember about this alpha ketoglutarate dehydrogenase complex. It requires all cofactors same as that of this pyruvate dehydrogenase complex. Remember this fact. It is not the isoscetate dehydrogenase complex, but the alpha ketoglutarate dehydrogenase complex. This is a very confusing thing. Okay, so the function, it converts the pyruvate ultimately into the acetyl coenzyme A, which can enter into the TCA cycle or the Krebs cycle. Okay, and now you should know about this arsenic thing, which actually inhibits the lipoic acid of this pyruvate dehydrogenase complex. So ultimately, it would inhibits your pyruvate dehydrogenase complex. So you won't get any conversion of pyruvate into the acetyl coenzyme A. So what would happen? All of this pyruvate will go and form the lactic acid, right? Because you require the NAD stores back to keep this glycolysis going on, right? So all of the pyruvate will convert into the lactic acid. So you will get the lactic acidosis, the high anion gap metabolic acidosis in it. And you can imagine something like a vampire. Okay, they will have this garlic odor. This is a very important thing. Please mark this garlic odor or the garlic breath. This is a buzzword you will always get whenever they want to give you a case of this arsenic poisoning. They will always have to mention about this garlic breath. You will get the vomiting. You will get this vampire thing, which is a skin changes, skin cancer like this xeroderma pigmentosum. Okay. You will get the skin cancer. You will get the blistering of the skin. You will get the ulcerations of the skin whenever you expose your skin in the sunlight, right? So this is arsenic. And they will ask you the treatment of this arsenic poisoning, which is the Saxaimer and the dimer-caprol. How can you remember these names? All of them name has R in it. The dimer-caprol has R, Saxaimer has R, and arsenic also has R in its name. So remember these drugs about the arsenic poisoning, and please remember this buzzword, the garlic bread. They will always give you that. Okay. And if you have this pyruvate kinase deficiency, which is an enzyme that converts your phosphoenol pyruvate into the pyruvic acid. If you get a deficiency of this enzyme, you will get this hemolytic anemia because you are no longer be able to continue your glycolysis. You won't get generate any ATP, and your RBCs will die because of it. So this is an important cause of this hemolytic anemia. All of our RBCs which will get damage into the spleen. So you will get the splenomegaly. You will ultimately get the iron overload. Okay, and because this step is inhibited, so what you will get all of the substrates before this reaction will increase. For example, like this fructose 2,3, sorry, this 2,3 biphosphogycerate. If you inhibit this step, the conversion of phosphoenol pyruvate into if you inhibit it, all of the substrates before it gonna increase, and one of them is a 2,3-biphosphoglycerate, which is a test that you will do to diagnose this hemolytic anemia due to the pyruvate kinase deficiency. Please remember this thing, the increase of 2,3-biphosphoglycerate. Okay, and you will study about Heinz bodies later. So pyruvate kinase deficiency, that's it. On this page, any doubt? Please
Sir,
respond.
why alanine have a inhibitor of 10th step of glycolysis as the mark?
Where?
In phosphenol pyruvate to pyruvate, alanine have inhibitor of this.
This.
Yes, sir.
Okay, so see, let me take you back again. You should know about these two. It's not clear. This is alanine actually, you see, this is alanine. And if you want your pyruvate to convert into the alanine, you need to inhibit it, right?
Yes.
This is alanine. Here you can see that. You said that later. Don't worry about that. It will come again later.
Okay, sir. Thank you.
Yeah.
Why do you have high anion gap though? You said it's because of the lactic acidosis, but I don't understand why. Or
okay, so if you inhibit your dehydrogenase complex, what will happen here? If you inhibit your dehydrogenase complex, then all of the pyruvate will convert into lactic acid, right? Right?
Yeah.
Lactic acid, and that's why you're accumulating extra acid inside your body. And whenever you accumulate extra acid inside your body, you will get the high anion gap metabolic acidosis. You will increase your anion gap because of this extra acid.
Okay, because otherwise it would have been just like the chlorine causing the normal, but this is more.
Yeah.
Thank you.
Yeah. Okay. Yeah, exactly. Okay, so now what will happen if you get a pyruvate dehydrogenase complex deficiency? I already told you the findings, the lactic acidosis, which is a high anion gap metabolic acidosis. As you can see, the pyruvate has 4 fates. Either it could be converted into the lactate, okay, which is the cause of this lactic acidosis. Either it will get converted into the acetyl coenzyme A, which requires this complex. So this step is inhibited. Either it could become the oxaloacetate, which requires a pyruvate carboxylase. Remember this enzyme, the biotin requiring step, and it is the first step in the formation of glucose, the gluconeogenesis. Or either your pyruvate will convert into the alanine. Okay, so if you inhibit this step, you will get high lactate and you will get high alanine. Okay, so serum alanine is increased, lactic acidosis you will get, you're not getting any energy, right? So the first thing that would be affected is your brain. So you will get the neurologic deficits. Okay, and you should remember that this is an X-linked, I think, recessive disease. Yeah, it is X-linked recessive disease, the pyruvate dehydrogenase complex deficiency. Okay, you should know the treatment of this disease. It is very important. You need to bypass this step. You need some substrates that can directly form the acetyl coenzyme A without forming the pyruvate, right? So these are there are two amino acids like lysine and leucine, which directly forms the acetyl coenzyme A bypassing this complete step. Please remember these two enzyme names. These are the ketogenic enzyme, and these are enzyme bol raha hu. These are the ketogenic amino acids. They cannot be converted into the glucose. As you can see, acetyl coenzyme A can never be converted into the glucose itself. Once the acetyl coenzyme A formed, it either can form the ketones or the fatty acids or the TCA cycle, but it can never be converted again back into the pyruvate. So these lysine and leucine, because they form the acetyl coenzyme A, they can never be converted into the glucose. So these are the ketogenic amino acids, and you are using them in the diet because they can directly form the acetyl coenzyme A bypassing this step. Okay.
And why we are giving these two amino acids? Only because they make acetyl-CoA?
Yeah, exactly. Because you need the TCA cycle to continue, right?
Okay.
That's why you're giving these two amino acids, because they can form the acetyl coenzyme so that you can get more energy in the TCA cycle. Because as I already told you, this step, the glycolysis will generate only 2 ATPs. Okay, so you need this step again, the TCA cycle. So the pyruvate metabolism, I told you, pyruvate has 4 fates. Either it can form the alanine or the oxaloacetate or the acetyl coenzyme or the lactic acid, right? You should know all these are transferase reactions for like alanine aminotransferase or the aspartate aminotransferase. They all require the vitamin B6. Remember the neurotransmitters and this ALT, AST thing, they all require this vitamin B6 as their cofactors. Okay. Similarly, the enzyme pyruvate carboxylase, which requires the vitamin B7, I told you all these carboxylase reactions require the biotin. And do you guys remember what the decarboxylation reactions required? Remember decarboxylation reactions?
Vitamin B6?
Yeah, exactly, vitamin B6. Please remember these facts. So you need this step to replenish the TCA cycle. As you can see, this pyruvate is being converted into the oxaloacetate, and oxaloacetate is one of the substrate in the TCA cycle. So you are actually using this step to replenish the TCA cycle, because whenever a acetyl coenzyme enters, it takes 2 carbon with it. As you can see, this one CO2 is removed and then another CO2 is removed. So neither there is a gain and neither there is a loss of carbon molecule happen whenever a cycle runs. Okay, so in order to replenish all these substrates, you need this step, the conversion of pyruvate into the oxaloacetate. Okay, then comes your pyruvate dehydrogenase complex. As I told you, it will convert it into the acetyl coenzyme. And finally, the lactic acid dehydrogenase, this LDH enzyme, which converts the pyruvate into the lactic acid, and it requires the vitamin B3. Okay, all these dehydrogenase reactions requires the vitamin B3 because they're involving this NAD and NADH. And you guys already know that vitamin B3 is the NAD and NADH. Okay. And you should know this LDH is a marker of cell destruction. Whenever a cell destruction occurs, whenever you get this, rupture of your membrane, what you will get is the leak out of this LDH from the cell. And whenever this LDH is increased inside your body, there must be some kind of a destruction going on inside your body. Whenever you get a myocardial infarction, you will get this high LDH levels.
Is that
Can
also
even see them?
iron
Yeah.
overload?
Yeah, please.
Is that also why you're getting the iron overload?
In which case?
In pyruvate dehydrogenase? No. So on the last page, pyruvate dehydrogenase complex.
It was kinase deficiency. It was this deficiency, this enzyme. Deficiency. It was different. Because no, this is a different case. Actually, in the hemolytic anemia, you are having this, the loss of RBCs. Your RBCs are getting destroyed, guys, right? And they contain this hemoglobin thing. Remember, and the heme contains this iron. So because of the destruction of RBCs, you are getting this iron overload in case of hemolytic anemia, and that's due to this enzyme deficiency. Okay.
Okay.
And this is a different case.
Mm-hmm.
This is a different case. In this case, you will having this LDH marker rise because of loss of plasma membrane, because of the cell destruction. Different thing, not the same. Okay.
Okay. Thanks.
So now we will move to the TCA cycle. Our time is going to run out in 3 minutes. So we can take a short break either, or after 15 minutes, we'll take a break. Okay. I will share a new link with you guys. Okay.
How much is the time of the break?
No, we will take after 6 p.m. Okay.
Okay.
Yes, sir.
We'll pause, stop the recording.